So, hi everyone. Welcome to day 2. And I hope you're all having a good time in Miami and enjoying the good food that hotel is providing for us. We're going to start this morning with a session on regulatory pathways. Peter Marks, who is the director of CBER at the US FDA, has kindly agreed to spend the morning with us. He's here virtually, and you will see him up on the slide. But he's going to talk about regulatory aspects of cell and gene therapy for rare disorders to start with. And then we're going to have a Q&A related to his talk. And then we're going to have us Ask the FDA session for questions. And we have some questions that people submitted in advance, but are also welcoming questions from the audience. So hopefully this is going to work like CNN. And I'm going to welcome Peter right now, and he's going to be right there on the screen. Hi, Peter.
Hi, hopefully everything's working.
Yes, we hear you fine, and we see you as well.
Okay, I'm going to go ahead and share my slides. Or someone's going to share them on your end, maybe. Let's see. I can't share my slides from my end here. Someone will either need to show them there or give me permission. That's all.
Okay, AV team. We have your slides back up, but I thought you were supposed to be able to share your screen.
Just need permission to be able to share slides, that's all. Or else if you need to show them there, that's fine.
I think they're working on it. Just give it a sec. Okay, they made you co-host, so you're good. I think you can.
And we're great. Thank you. Sorry about that. We should have done that to start. There we go now. Okay, and now you probably can see my slides. Thanks a lot.
We do.
Okay, I'll talk about regulatory pathways for cell therapies for very rare diseases. What very rare means is a little bit in the eye of the beholder. I will tell you that what I mean by very rare are diseases that affect tens to hundreds of individuals globally, not thousands to hundreds of thousands. So some people call these ultra rare. That gets into a whole nother issue. But what we're talking about are diseases which don't affect a lot of people. And I would actually say that almost the operational definition of these diseases these are ones which currently, under our current regulatory environment and our current manufacturing environment, development environment, are not really commercially viable. So that's another kind of operational definition instead of a numeric definition. Just by way of full disclosure, I work for the US government and they don't have any relations with commercial interests to disclose. In fact, they don't have any relations with commercial interests that are financial. So I'll divide this talk really into 2 major portions and then a minor one. The first part about our efforts at FDA to facilitate the development of cell and gene therapies. These have taken a little bit of a backseat during the pandemic, and we're now really coming and moving forward again full speed in this area. And we'll then talk a little bit about the applicable regulatory framework for cell and gene therapies. And at the end, talk a little bit about resources for product developers. So just the bottom line here is that we are very committed to advancing development of cell and gene therapies for populations of all sizes, but particularly this area of rare diseases, we think, as I'll explain a little bit later, is one that is important to concentrate on because it may serve essentially as the lab in which we get things right for larger populations. We hope to be able to move development forward by individualizing product development, by coming up with the right types of clinical trial designs and really thinking about how we focus on manufacturing. So just to say that during the past years, we've witnessed continued growth in these areas. And if you look here, you might think that in 2021, we had a drop off of investigational INDs. But if you realize we were in the middle of a pandemic, and you also realize that we had a fair number of expanded access requests during this time for INDs. The number of investigational drug applications has grown every year. And the panel on the right, which just shows the IND amendments, all of our IND amendments, shows the continued growth in this area. I actually think that probably it's actually remarkable that during 2020 and 2021, there was as much growth as there was, given how challenging it was to actually get things moving during that time. What this has all resulted in is the fact that we now have 9 approved gene therapies. Granted that 6 of these are cell-based gene therapies, but we now have besides the cell-based they're really CAR T-cell therapies, we have a directly administered gene therapy in Luxturna for a hereditary retinal disorder. We have Zolgensma, which is for spinal muscular atrophy as a systemically administered gene therapy. And then we have a genetically modified stem cell therapy, Zynteglo, which is just very recently approved. And we have a pretty robust pipeline of things coming along in the next year or two. So I think this list will be growing. I think the onasemnogene or Zolgensma is a good example of kind of a boundary condition of why these gene therapies are so important. Spinal muscular atrophy is one of the more common rare diseases, so it actually is commercially viable. Spinal muscular atrophy type 1 has a relatively typical progression in which children are born looking normal, but within a few months it becomes very obvious that something is wrong. They become floppy infants, can't hold their heads up, etc., don't make developmental milestones, and they deteriorate to the point of being on ventilators and succumbing to the disease, usually by 2 to 3 years of age. There was an oligonucleotide-based therapy for this that had to be administered repeatedly, and then this gene therapy was developed. The gene therapy, when it was developed, as published in the New England Journal, you can see on the left here, 14 out of 15 of the children that were treated essentially developed normally. The dotted line that's horizontal there, if you're below it, you're abnormal on the Children's Hospital of Philadelphia in 10 score. If you're above it, you're developing normally. And you can see that 14 out of 15 kids going out to about 2 years post-treatment were doing normally, which really remarkable. Doesn't require a statistician to say that something is fundamentally different with this gene therapy than it was before these kids got this. This is one of those times when there's the beauty of gene therapy is the genetic makeup of the child, you know the intervention you've done, and if you see something that's as remarkable as the child on the right running around at age 3, you know something is different. And if you see 14 of those children running around, you know that you have something that is quite remarkable. So this is kind of, I think, the poster child case of what we'd like to see happen. We know that development is more challenging for many diseases than this one because of the nature of many of the diseases that we're addressing, where they are more gradual progression where endpoints can be more challenging to assess, or where natural history is less well understood. But nonetheless, these very rare disorders are really important because even though, as I've said, each one may only affect, tens to hundreds of individuals, when you take in aggregate the thousands of these hereditary acquired rare diseases, you can put together the fact that you've got millions of individuals affected. And so addressing these, many of which can be, they're debilitating and or fatal, these do make a difference in human health. If we could address them, it would make a difference. Additionally, if we could get it right in addressing these rare diseases in a facile manner, we would be able probably to do gene therapy for much larger populations. And we would have a good sense of the safety of gene therapy for much larger populations, because there could be great benefits to treating certain things with gene therapy in the long run, even more common conditions, some of which aren't life-threatening. But you'd have to know that you had the safety and the efficacy to make it worthwhile. And so this is a really good, it's a good starting place, because in general, the benefits in these populations greatly outweigh any risks. And that's kind of the calculus that goes into this. So I just want to digress for a moment to say that I view this as really a kind of a transitional moment where, personalized medicine is something that I grew up with as a resident and fellow in hematology oncology, where we were looking at biomarkers in individuals and then pulling stuff off the shelf that addressed those biomarkers. And that was personalized medicine, right? You get flow cytometry results and on a lymphoma, and you'd know whether or not you could treat with certain monoclonal antibodies. Personalized medicine. And now, though, we're moving to individualized medicine, where instead of just looking for something off the shelf, we're making things to treat the patient. And I think that is an exciting place to be. It has also, though it's exciting, it has its challenges associated with it. Really, it's a transition from the world of essentially ready-to-wear of personalized medicine to individualized medicine, where we're either doing things that are made to measure, that is, we're kind of customizing products, or it's bespoke tailoring, where we're creating them from scratch. And what I mean by that is that customized products are things like autologous chimeric antigen receptor T cells, or personalized cancer vaccines, where you take the tumor from an individual and then make a vaccine out of that using a cell line, or either autologous or allogeneic. And then there are created products where we basically start from a vector that might be used for different diseases. We take that vector and put in different inserts to create products for small groups of individuals. The issue for these things is that we really have, to date, had challenges moving things forward. The challenges moving forward have come in the areas of manufacturing, non-clinical development, also known as preclinical development, or animal models sometimes, or manufacturing, and then product access. And although access isn't FDA's bailiwick fully, it is indirectly, in that we are very aware that if the costs of making a product are very high, it tends to reduce the amount of access. So we understand that if we can help facilitate development at reduced cost, that's a good thing. So we've been trying to address various things, including the challenges in non-clinical development, that is, working to find better animal models, such as humanized mice, or organoids. We've been working to find better ways of doing clinical development. We have a statistical group that focuses on Bayesian statistical designs, Bayesian design trials, where basically it's almost a continual reassessment model where each patient that enters a trial contributes information to understanding the size of the patient population you need to treat and the success criteria, such that really what you're doing is instead of starting out with a population size that you need to treat, you figure that out as you go. And then the manufacturing issues are ones that have been quite significant. And we're trying to work on that by doing several things. One of the challenges is, as those of you in the room may well understand, many of you go out and work on these rare diseases, and you have the patient population. But there really isn't a great playbook to follow in how you should go do this. So many people rely on what some other investigator at their institution has done, or a protocol they get sent to them from someone. Although it is very useful for getting started, it can create challenges because when one goes to move that technology over to for instance, a contract manufacturer, the fact that so many different methods of doing this are being used means that it's harder to transfer to contract manufacturers. So one of the concepts we've been developing is whether we can develop some essentially uniform protocols for the development and manufacturing of these bespoke therapeutics, these products for small populations of individuals, because if that transfer to, for instance, contract manufacturing organizations could occur more easily, it would reduce the cost. When they transfer something that they have to do a lot of rework for, they charge for that rework. If that rework didn't have to happen, that would hopefully remove that. Additionally, for those products that really show promise early on that a commercial partner would want to license, having something that had been made using kind of an established recipe might add value. And so it might actually also bring benefits. So it's kind of a two-way street. It could potentially bring value to the investigator institution as well as to the receiving entity. And then we're really also trying to figure out what we could do in terms of FDA's regulatory framework to make things easier in this area. And that has to do with really understanding that these gene therapies, whether they are viral vectored or non-viral vectored, have certain things in common that differentiate them from more traditional small molecule drugs. That is that gene therapies have this backbone generally when we're using them. That backbone is the vector, whether it be a viral vector or a non-viral vector. Right now, the one for example right now, I'll talk about adeno-associated virus because that's the most common vector being used for directly administered or locally administered gene therapies. But we could use other examples. But if you think about it, the backbone of the vector, the guts of it, so to speak, are often the same from one serotype AAV to the other, with different inserts being placed in. Now, granted, one has to make sure that the inserts stay within a certain size range, but the idea here is that that vector is more like a scalpel blade in some ways than it is like a small molecule drug, in as much as it can remain the same with different inserts, scalpel blades, inserted, and that totality then is the therapeutic product. So right now, each time we come along, we treat this as a new entity. So we don't leverage the similarity between the different vectors or the vectors that's the same, essentially. And so the idea here is if we were to be able to start doing that, maybe we could speed things up because we wouldn't have to redo the toxicology, we wouldn't have to redo the manufacturing description every time, and you could possibly have essentially one prototype product that, or originator product that then was followed along. And so the idea here is for these bespoke therapies to try to potentially have an originator product, and I'm deliberately using terminology that's very weird here. And the reason why is I'm trying not to use any terminology that's going to get me into legal trouble, because it says it actually steps on something else in our Code of Federal Regulations. So these terms are a little bit hokey to avoid getting into things that might step on the feet of generic drugs. But the idea here is if you had a kind of an originator product, a well-characterized product where you understand the vector well and the insert is placed in that, that you could come along then with several other products based on that same vector with an insert of approximately the same size, addressing different diseases, and you could reference back to that originator product rather than having to go through the non-clinical and manufacturing descriptions again. That saves time up front, but it also saves time out back, because the idea here would be then that we know that over the course of years, oftentimes there are advancements, improvements in how we manufacture gene therapies. And if you had this series of linked products, one would only need to go back for that originator product, change that application, and then you would essentially have that filter down into the other applications without having to submit the supplement umpteen times for the offshoot product. So this is an idea that we're working on. It's not fully hatched yet, but it is certainly taken on some legs. Congress is discussing it as a possible pathway to support. So we'll see where this goes, but I think it's very attractive, again, because the issue here is if we want to deal with hundreds to thousands of rare diseases, we really have to find a way to expedite the development pathway for very small numbers of individuals, because otherwise there'll never be a commercially viable way to make this happen. And without getting into the economics or the politics too heavily, the issue here is that unless there's a commercially viable pathway, it's hard to see how we will be able to address all of the different rare diseases, because it turns out that commercial viability is the great equalizer in terms of getting products made for a wide variety of different individuals. One of the ways that we're seeing this kind of in a testing realm is in the Bespoke Gene Therapy Consortium, which is a venture from the Foundation of National Institutes of Health, in which now a variety of different commercial entities and nonprofit organizations have been brought together to try to essentially test out whether what I just presented could work in the real world. In other words, you standardize manufacturing, standardize vectors, and use all of the tools we can to try to study small numbers of individuals and come to the clinical data that would be necessary to get to an approval, either a traditional or an accelerated approval. And so hopefully in the next few years, we'll see this bear fruit. So I want to move now to talking a bit about FDA's regulatory role. The FDA is comprised of a variety of centers. We have a pretty large remit, including medical products, which are regulated by the Center for Biologics for more complex biologic products, including blood products, vaccines, and cell and gene therapies, as well as xenotransplantation. Center for Drug Evaluation and Research takes care of small molecules, homeopathic drugs, and also many of the therapeutic biologics that are hormones or monoclonal antibodies. and Center for Devices and Radiologic Health, which takes care of exactly what it sounds like. And then we also have, obviously, we deal with foods and tobacco as well as veterinary medicines. As an aside, veterinary vaccines are regulated by the USDA. Interesting little side note there for trivia. Our regulatory framework for biologics dates back to before the FDA was established in the Biologics Control Act of 1902, which basically gives us all of the authorities that we still have in the Public Health Service Act, Section 351, which is what allows us to license biologic products in the United States. Back in Teddy Roosevelt's day, somebody in just 3 pages of text managed to summarize actually an awful lot that made sense, which is that for biologic products, we have the ability to make sure they're safe, pure, and potent. They gave us licensing authority. They gave us the authority to revoke licenses, and they gave us the authority to inspect so that we would know whether to revoke licenses. And actually, those are things we hold dear in the biologics world because we don't have those for all other medical products. Obviously, the Federal Food, Drug, and Cosmetic Act came later and added some additional aspects to this, but largely what we deal with right now for biologic products like cell therapies comes from Section 351 and 361, which came later of the Public Health Service Act, and I'll say a little bit more about that. For those who aren't familiar with FDA speak, you know, the laws and statutes that govern FDA are very high level. As I've told you, the original Biologics Control Act of 1902 was just a couple pages. It gave us broad authorities, but it didn't really say how we were going to go about doing those things, and didn't give a lot of information for those who were developing products. And so what we have, and as is the case for many government agencies, is based on the laws and statutes. We do rulemaking, which is done by a process called notice and comment rulemaking. We say we're going to make a rule, then we put out a draft of the rule, people comment on it, we answer the comments, and ultimately the rule goes final, and those rules are then collected in the Code of Federal Regulations, which for FDA is 21 Code of Federal Regulations. But there's more because the regulations take so long to do because of that process of saying you're going to make a regulation, proposing it, getting comments, et cetera, that usually takes years to happen. So on the fly, when we need to say things quickly, like during a pandemic, when we need to tell people how we're going to go about making vaccines, we can issue guidance. And guidance does not have the force of law that regulations do, but it is the agency's best thinking about how to do things according to our regulations. And so when you hear that term guidance, that's what it means. So the other piece of this is that over the course of years, either through rulemaking or through Congress giving us laws, we have a variety of expedited development programs which are relevant for developers. Fast track priority review and accelerated approval were ones that were around. And then in 2012, they were wrapped up together along with some additional benefits like having additional meetings and having senior leadership involved in development in breakthrough therapy. And so breakthrough therapy has been something that has become a useful tool for products that show promise early on, or not so early on, as significant advances over existing therapies. And then more recently, at the end of 2016, we were given the authority for regenerative medicine advanced therapy designation, which is really breakthrough therapy, especially tailored to cell and gene therapies. All of these obviously are for serious or life-threatening conditions. That's where the agency feels like it's worth spending the extra effort. Now, we put out in 2017 a suite of regenerative medicine guidance documents to help clarify our regulations and make it easier for people to understand what things needed to come in for licenses and what things did not need to, because there are certain tissue products which are simply regulated under provisions that help prevent the transmission of infectious diseases. Those are affectionately known as 361 biologics. They're regulated mainly under the provisions of the Public Health Service Act that that allow us or direct us to prevent the transmission of infectious disease either interstate or into the United States. But we also wanted to try through these guidance documents to talk about how people could more facilely develop, how they could more rapidly develop and more easily develop regenerative medicine products. And so we have a suite of guidance documents that came out then that are now all final. One on the same surgical procedure exception. That's basically when you actually don't even need to even get involved with the infectious disease transmission issues. We have an important guidance on understanding when something is minimally manipulated and homologously used, because those products can potentially fall under our tissue rules. We have a guidance that was on how to make devices for regenerative medicine, advanced therapies, and one that I'll tell you a bit more about on our expedited programs for regenerative medicine therapy, particularly how we apply the RMAT designation. So really the expedited programs for regenerative medicine therapies guidance really talked about how this RMAT guidance, which is the acronym we use to describe it, applies to certain cell therapies, tissue engineering products, human and cell tissue products, and combination products. It also noted that we interpret this as also applying to genetically modified cell therapies and gene therapies that produce durable effects. And so all of these fall into this category as being eligible for the designation, which, as I've noted, has to be for serious or life-threatening conditions. There has to be preliminary clinical evidence that they can potentially meet an unmet medical need. It's not, though, that it has to be better than a standard of care, okay? So that's a little bit of a lower bar to get the designation. Additionally, if one gets the RMAT designation, there is an expanded way of fulfilling post-approval commitments if one gets an accelerated approval. It was understood that for some of these products, up front, it was understood that some of these products might be for very small populations. And so the traditional way of doing a second confirmatory trial in a larger population, potentially, to confirm the benefit, clinical benefit from an accelerated approval, was realized that might be a challenge. So the provisions allowed, for instance, for taking the individuals who were studied as part of the initial accelerated approval, following them for a longer period of time, looking at a clinical endpoint, and using those data to convert over from an accelerated to a traditional approval. So a little bit more flexibility here. If we look, this pathway has been used. We're getting close to 200 requests. And as of June 30th of this year, we had granted 77 RMAT designations since the program was started. Many of these are cell therapy products. Many of them are for relatively uncommon diseases. These numbers here is essentially about 40% granting rate for RMAT designation, mirrors that for breakthrough therapy designation. So there is some parity here with what's going on in small molecules and other biologics. The regenerative medicine advanced therapy designation has actually eclipsed breakthrough designation in terms of what we receive as requests and grant. It's not surprising, because if you can receive a regenerative medicine advanced therapy designation, since it has those the slight advantage in the accelerated approval endpoints, there's no reason not to. Sponsors seem to realize that. But some actually apply for both regenerative medicine advanced therapy designation and breakthrough therapy designation, you can do that, but it's really redundant. But again, all things just can be done. I just want to also tell you that in addition to the regenerative medicine advanced therapy guidance in this space, we've recently issued some additional guidance, on the consideration for development of CAR T-cell therapies. We think this is something that's important since these therapies are becoming more widespread. We're currently the autologous CAR T-cells are becoming established, certainly very well established in hematologic malignancies, but are starting to expand into solid tumors. And we're starting to see more and more allogeneic CAR T-cell therapy investigation there because particularly using the genome editing, one can make a sufficient number of edits to modify the cells so that they can address multiple targets, which is really what you need to do in many solid tumors because you can't find a single antigen to target. Whereas if you can target 2 or 3, you can actually potentially have a therapeutic CAR T cell. So we'll see more where that all goes. But this guidance kind of talks about the considerations for making those CAR T cells and manufacturing them too. And then we have I mentioned genome editing. We put out a draft guidance on human gene therapy products incorporating human genome editing. That's given, although genome editing has been around for a while based on other systems for editing the genome, CRISPR-Cas9 certainly moved that forward significantly. So we'll see, just people can see the agency's thinking there, and we welcome comments on that. Just to finish up, we do have various ways that people can get feedback from the agency. We have an advanced technology team which really focuses on manufacturing, particularly of platform technologies, cell lines, manufacturing methods, vectors, novel platforms. And for those who would like to come in and get kind of informal, it's not on the record, non-binding feedback about this, their products or their platform, this is not for someone who has a specific product in development, but rather who has a platform or a series of products that are a platform. It's really a nice opportunity for a sponsor to get some feedback. It's also a nice opportunity for us at FDA to learn about novel technologies. And these generally are pretty easy meetings to prepare for because we only require a slide deck, a couple of questions on the slide deck, and we usually have been having hour-long virtual meetings during the pandemic on these areas. And I think both sponsors and FDA has benefited. For products where it's a specific product that someone is developing and they want more information, again, in an informal setting, not even with the formality of a pre-IND meeting, we have the Interact program, which allows sponsors to come in and ask, again, their non-binding advice. We don't hold anyone to it, but based on, it requires a little more write-up, perhaps. We usually like to get a write-up of what's the product for, what are the thoughts about development, and what are the questions. But with just a little bit more information, we hold these meetings. And our website, if you search up Interact and CBER, describes how to get one of these meetings and what you need to do for that. But this, again, is a way of getting early feedback. We really have come to realize that getting things right earlier on in development is really helpful. And I think in having done this from both the industry side of things and the FDA side of things, as well as actually even in the academic side, it's like many other things in life. If you start with a good foundation, it's much easier to build the house. And so we view this as essentially the equivalent of building a good foundation. So with that, I'll just conclude by saying that we really are committed to advancing the development of cell and gene therapies. You will see that commitment over the coming years. I think stay tuned, though I can't go into the details right now, stay tuned for some announcements over the coming months about additional change in this area at FDA to try to facilitate the development of cell and gene therapies in a more facile manner. Certainly, it's already public that as part of the next round of Prescription Drug User Fee Act, whenever it's passed, we will likely get a significant wave of staff added in this area, which should really help us be able to give the type of feedback in a timely manner that we'd like to. Right now, although I think we know what we want to accomplish, we're not able, because we don't have enough staff, to be able to do what we want to do in a timely manner, and that's really critical. So hopefully that will help there. So a lot moving in this area, and I'm really encouraged that hopefully coming out of this pandemic, we'll make a lot of progress in this area to see more and more gene therapies become a reality. So I'll stop there. Thanks very much.
So thank you, Peter. Thank you, Peter. I hope you could hear everyone applauding. And we're going to go first to Q&A for your talk and then ask the FDA session. But I'd like to invite my colleague Beth Shaz to come up and join me. I'm staying on the podium so you can see me on my Zoom and not be talking to no one. But are there questions from the audience for Peter in reference to the talk that he just gave? If you have a question, come up to the microphone, please. Well, I have a question. It's a little wonky, but I was really interested when you were talking about the originator and the offshoot concept. And I wonder if that can be applied to cord blood, which publicly is a licensed product, and offshoot products. I'll use us as an example, like using cord blood to treat cerebral palsy, where an entity that has a license could join in clinical trials, not change manufacturing, but show efficacy in clinical trials for a different indication. With the hooker being, or the elephant in the room being, is that homologous use or not? And, cord blood was approved for hematopoietic reconstitution after myeloablative therapy with the potency being CFUs or a CD34 cell. That really isn't the way we think cord blood is working necessarily in cerebral palsy. So how does all that get reconciled? And is there room in this concept to think about that?
Yeah, so I think I'd like to, I'd separate out two pieces. There's the clinical data that would go into an approval, which would probably have to be, each indication would have to stand on its own. There, you'd have to have that. I do think there might be room to think about how the manufacturing pieces of these could rely on one another, essentially, because you basically could potentially rely on the same manufacturing filing here. The major issue to date that we've seen with this actually is that entities have to agree to share things. And we've seen people sometimes not want to share in this area. But I think we actually, even our current framework allows for this. So that if you are using essentially a product that's manufactured by an identical manner, I think we could probably accommodate that.
Okay.
By right of reference. Obviously, you have to have right of reference, and that's where sometimes things get tricky.
And I think, I'll speak for the people in the room, we're a sharing community. Roberto.
Yes. Hi. Some companies say that in the past, say 10 years ago, the FDA issued approval for what we call today cell therapy products. And as an example, cultured autologous fibroblasts. Did the FDA actually revoke these authorizations? Were they ever issued at all?
It's really interesting. I don't know that we ever authorized cultured autologous fibroblasts. It's possible that what happened was there were people who interpreted certain products to be covered as those 351 products. They interpreted them as not requiring biologics licenses. And there's still a whole bunch of things out there that, because it's a self-determination, people have products that might really be more appropriately regulated under a biologics license application that they are essentially selling as, end quote, in compliance with FDA regulations, because they've made the determination that they're not a product that needs licensing, but that does not mean that the FDA has made that determination. And sometimes they usually get by until something bad happens, and then when we go out and investigate, that's sometimes when people get into issues where we have to tell them that they should have had a biologics license. The hope is, honestly, my hope is that over the coming years, we can find a place where that some of the products that are in this space, where I think it would be helpful if we had some more data on in terms of what they are, in terms of efficacy. Some of these, the safety isn't the issue. It's that is there efficacy for the intended use, that we can find a way towards getting those data in a simple enough manner that's not too burdensome, so that people will come in with those data, and then we could give, we could find a way to give an approval of some type, so that one would actually have the appropriate reimbursement for products. Because right now, there are many products that people are just paying out of pocket for, because insurance companies, just as FDA, we don't know that they're effective or not. Insurance companies say the same thing. And so it would be nice to try to get those in with some data.
Thank you.
Great. Well, I don't see any questions from the audience about your talk, any more questions. So we'll move on to the section of the session we're calling Ask the FDA. Thank you, Peter. I hope you could hear everyone applauding. And we're going to go first to Q&A for your talk and then an Ask the FDA session. But I'd like to invite my colleague Beth Shaz to come up and join me. I'm staying on the podium so you can see me on my Zoom and not be talking to no one. But are there questions from the audience for Peter in reference to the talk that he just gave? If you have a question, come up to the microphone, please. Well, I have a question. It's a little wonky, but I was really interested when you were talking about the originator and the offshoot concept. And I wonder if that can be applied to cord blood, which publicly is a licensed product, and offshoot products. I'll use us as an example, like using cord blood to treat cerebral palsy, where an entity that has a license could join in clinical trials, not change manufacturing, but show efficacy in clinical trials for a different indication. With the hooker being, or the elephant in the room being, is that homologous use or not? And, cord blood was approved for hematopoietic reconstitution after myeloablative therapy with the potency being CFUs or a CD34 cell. That really isn't the way we think cord blood is working necessarily in cerebral palsy. So how does all that get reconciled? And is there room in this concept to think about that?
Yeah, so I think I'd like to, I'd separate out two pieces. There's the clinical data that would go into an approval, which would probably have to be, each indication would have to stand on its own. There, you'd have to have that. I do think there might be room to think about how the manufacturing pieces of these could rely on one another, essentially, because you basically could potentially rely on the same manufacturing filing here. The major issue to date that we've seen with this actually is that entities have to agree to share things. And we've seen people sometimes not want to share in this area. But I think we actually, even our current framework allows for this. So that if you are using essentially a product that's manufactured by an identical manner, I think we could probably accommodate that.
Okay.
By right of reference. Obviously, you have to have right of reference, and that's where sometimes things get tricky.
And I think, I'll speak for the people in the room, we're a sharing community. Roberto.
Yes. Hi. Some companies say that in the past, say 10 years ago, the FDA issued approval for what we call today cell therapy products. And as an example, cultured autologous fibroblasts. Did the FDA actually revoke these authorizations? Were they ever issued at all?
Really interesting. I don't know that we ever authorized cultured autologous fibroblasts. It's possible that what happened was there were people who interpreted certain products to be covered as those 351 products. They interpreted them as not requiring biologics licenses. And there's still a whole bunch of things out there that, because it's a self-determination, people have products that might really be more appropriately regulated under a biologics license application that they are essentially selling as, end quote, in compliance with FDA regulations, because they've made the determination that they're not a product that needs licensing. But that does not mean that the FDA has made that determination. And sometimes they usually get by until something bad happens, and then when we go out and investigate, that's sometimes when people get into issues where we have to tell them that they should have had a biologics license. The hope is, honestly, my hope is that over the coming years, we can find a place where that some of the products that are in this space, where I think it would be helpful if we had some more data on in terms of what they are, in terms of efficacy. Some of these, the safety isn't the issue. It's that is there efficacy for the intended use, that we can find a way towards getting those data in a simple enough manner that's not too burdensome, so that people will come in with those data. And then we could we could find a way to give an approval of some type, so that one would actually have the appropriate reimbursement for products. Because right now, there are many products that people are just paying out of pocket for, because insurance companies, just as FDA, we don't know that they're effective or not. Insurance companies say the same thing. And so it would be nice to try to get those in with some data.
Thank you.
Great. Well, I don't see any questions from the audience about your talk, any more questions. So we'll move on to the section of the session we're calling Ask the FDA. Great. Well, I don't see any questions from the audience about your talk, any more questions. So we'll move on to the section of the session we're calling Ask the FDA. And we solicited you all to submit questions, so we have some questions submitted that we'll start with. But if others have questions that you didn't submit, we still encourage you to ask, and we need you to come to the microphone to do that. But while people are thinking, I'll have Beth start with one of the questions that was already submitted. Actually, it's a question that we got more than one submission on, so it's a burning question for the community.
Okay, great. So question number one. Given the lack of current Zika outbreaks since 2018, no evidence of Zika transmission by HCTPs, and FDA's determination that Zika was no longer a relevant transfusion-transmitted infection, resulting in the withdrawal of the revised recommendations for reducing the risk of Zika virus transmission by blood and blood components, which was issued in July 2018, will the FDA be updating or withdrawing the donor screening recommendations to reduce the risk of transmission of Zika virus by HCTPs, which was issued in March 2016 and updated in May 2018?
So this is one where I wish I could say more about the content, but what I can tell you is it hasn't escaped us that this needs updating. And there will probably be some updates to a variety of the donor eligibility criteria for tissues coming this fall and into next spring. It probably hasn't escaped some that our blood donor and our tissue donor regs, the guidance is different right now, and over the course of the next year, we intend to harmonize more and more of this. And in the process of doing so, I suspect that you will see us address things like Zika as well.
Well, we are thrilled to hear that.
Great news.
All right, Joanne, we'll do the next question.
I'm going to go out of order for a minute. A related question that came in was, is there any consideration being given to harmonizing the donor screening testing guidelines with the EU, which I'm sure you know is also updating some of their regulations?
A really great question, and the best I can say is we're having conversations there. There actually are some, it's just challenging sometimes with the EU, because not only do we have the EU as a whole to deal with, we have the individual member states to deal with at times, and EMA often is not, there's often not full alignment between necessarily EMA and each of the countries, and it's a slightly different method of regulation. All that to say, though, that we are trying to work as best as we can here, and we have a variety of groups that work in this area to try to harmonize. Whether we'll achieve it or not, I can't promise.
We have a comment from the audience.
Gesine Kögler from Germany. It's not so much about what kind of testings we do, because this is quite harmonized in the European Union. I mean, Germany is special, we have to add other testings. But it's also the general questions that for the FDA, usually the European donors are still excluded or marked based on the situation that they were living in Europe based on the BSA. I mean, this is strange because also people from UK travel to Australia. So, and then, I mean, this is a history of 1980 to whatever, but there is still marked, it's still marked coming from the commands from the FDA to European companies or to European products that they are tainted somehow.
So this is actually excellent because it's completely related to the last. So the guidelines for transmissible spongiform encephalopathies is one of those other guidances that is currently not harmonized between blood donors and tissue donors. You'll note for blood donors, people from Europe are not marked anymore. And in fact, we've gotten rid of this because
great.
It turns out that we're not really worried about this issue anymore. You're exactly right. We're 30 years out now, and we're not seeing secondary transmissions that people were worried about. So we've caught up in the blood donor world. So we now have to catch up in the tissue donor world. And again, that's I would stay tuned over the course of the next number of months so that we can deal with that, because that will, I think, help bring more harmony there.
Okay, thank you.
Yeah, that's good news too. I know you know, but I'm just going to say out loud that in the cord blood and related cell therapy businesses, these products cross the ocean a lot. In the transplant world, these products cross the ocean a lot, and it's either best donor or autologous donor, whatever it is, there's good reason for the products to cross. So making that more harmonized would be wonderful. Just a comment.
Got it.
Next question, completely different subject. There are many supply chain issues related to cord blood banking and lots of other things in our world, as you know. But in cord blood, these include shortages of collection bags, processing kits, thawing kits, and reagents that are used in processing or thawing, like Hespan and Dextran. In addition, as a recent example, a company called Cytiva, which makes an automated device for cord blood and other processing, announced that they'll be exiting from the cord blood device manufacturing business in 2026. And they're taking many steps to restrict manufacturing of devices and kits starting in 2023, which is next year. This affects a device called the CPAX, which is used for red cell and plasma depletion and volume reduction of fresh cord blood units in an automated fashion, volume reduction of bone marrow and peripheral blood and mononuclear cell collections, thawing and washing of HCTP products, and the associated kits for these procedures. These devices and kits are currently validated and used under many banks, BLAs, or other regulatory approvals in the USA and other countries, and obviously will create great challenges for the banks that have BLAs based on this kind of processing. Furthermore, Cytiva plans to continue to produce the research devices produce the devices as research devices, but not medical devices that will perform similar or identical functions. They'll also produce kits that perform the same function, but under the research use only designation. So will the FDA allow cord blood banks or other HCTP manufacturers to validate these research devices and kits to enable continued use of the devices and kits under their BLAs or similar regulatory approval?
Yeah, no, thanks for that question. It's a very good question, because this is another one of those cases where FDA in shortage situations has generally worked with manufacturers and with the communities to try to find ways to address these, sometimes relatively novelly, by making various products available under various pathways. I think this is one where it seems to me, Joanne, that it makes a lot of sense for, in talking to our compliance folks, for someone to come in sooner with Cytiva rather than later, to have a conversation with the agency about what we could allow in this setting, which is a potential shortage situation. Better head it off at the pass rather than waiting for there to be a shortage situation, because there may be a variety of ways to deal with this. Some of it actually may depend on the nature of the product that's ultimately being produced. In some cases, from talking to at least our attorneys, it may turn out that since it will be something that will be considered in the manufacturing process, it actually wouldn't be a problem even now, whereas in other cases it might be. So it's probably worth the conversation, and I wish I could give you the full answer here, but I think the best thing that we would say is that we would be very willing at the agency to have a conversation about how to avoid this problem, which, by the way, is one we've dealt with all too often in the pandemic. Yeah. In fact, it's very funny because yesterday I was talking to a group about manufacturing during the pandemic, and there was a point early on in the pandemic where I felt like I was the manager of plasticware for the United States government, because we were like, we didn't have enough 500-liter bioreactor bags, and at one point blood collectors were running out of pipette tips. So
right.
It's a problem. We realize that.
Yeah, there's a comment from the audience.
Yeah, I have a question, because you showed in your presentation that, for instance, for validation of viruses or whatever, that there could be a common platform. That means if one lab, for instance, is submitting a file to the FDA for processing, and we are all doing the same processing, for instance, yeah, it's quietly regulated. Could it be of use that not everyone has to do or file it again?
Right, no, great. It's a great question. And so the way we have, our way of getting there, which is what you'd be saying is kind of the right of reference for a standardized technique.
Yeah.
Is we feel like, because we don't have a lot of experience with this, the first step we'll do is allow a given manufacturer to do that with its own products. And once we get some experience that, where we see that that's working really well, that nobody has problems with their own products, I think we can start to think about manufacturers going across, from one to another. There is a peculiarity in our U.S. law that means that we may need some modifications to our laws to make that happen, but it certainly makes a lot of sense once we feel comfortable from the scientific perspective. Again, you're totally right? If something can be done similarly, we shouldn't be making people, I think we all agree, right? There's only so many hours in the day. Why reinvent the wheel if the wheel is working well?
Okay, thank you.
So kind of extending that question, we're contract manufacturers for BLA, and we're experiencing supply chain issues around media, around vessels, around containers. We know there are equivalent containers, but the original process that had to be done to validate container closure and leaching and all those kind of things in a quick situation can't be repeated again. So how much latitude would FDA give in those kind of settings to enable switching to a different source if the source can be shown to be compatible by the testing of the manufacturer?
So this is one where this is one where a discussion with us is in order. I hate to constantly fall back on that,
no,
but
that's okay.
It's really case specific, because you just noted it. If you can show, if we can feel with a straight face like the media, for instance, a media is interchangeable, or that there's no, we've had this happen in the gene therapy world, where the company making this sole company making a transfection reagent went out of business, and then you were left with, you had to switch to something. We're happy to have those conversations, and as long as we feel like we, with a straight face, can say that the switch can be made, sometimes we might ask for data on the back end. We're happy to have those conversations.
Great, thank you. Okay,
All right.
Switching topics.
Okay, we're going to go on to monkeypox. We thank the FDA for issuing the guidance on monkeypox and HCTP donor eligibility. This includes information regarding donor eligibility based on receipt of different smallpox vaccines. We have the Junis, which is a replication incompetent virus, versus the ACAM 2000, which is an attenuated vaccina-based orthopox vaccine. We wanted to make sure that if anyone in the audience had questions for Dr. Marks about this information. It wasn't issued as a guidance. I don't know if that wasn't really listed as one of your options on this screen, but
let me just say a few words, well, and maybe we can have an interactive conversation about this. So ACAM 2000 is a descendant of Dryvax, which was the smallpox vaccine that was used when we really were in the heyday of vaccinating against smallpox. Dryvax is and ACAM 2000 are live replicating vaccinia vaccines. They produce their effect after being given essentially percutaneously. You give them with a bifurcated needle with 15 passes under the skin. They leave if things work properly, you get a nasty looking scar. I will save you. I will save you looking at mine. You end up with a nasty.
Absurd. Okay.
And then during that time, the reason we're not thrilled with using that currently because, especially around immunocompromised individuals, one sheds the vaccinia, and it actually can cause problems in immunocompromised individuals. The other problem with ACAM 2000 is that it's associated with about a 1 in 175 individual incidence of myocarditis, and it's an immune-mediated myocarditis. So when you have a threat like smallpox, it's very clear that the benefits outweigh the risks. When you have monkeypox, where really the fatalities that have occurred globally are a few handful, and it's mostly in really immunocompromised individuals, that's why we've tended to use the Jynneos vaccine, which is a non-replicating modified vaccine, Ankara. A shout out to Germany, where it was developed in the early 1970s by Professor Stickel, who gave it via the intradermal, and colleagues who gave it via the intradermal route there. It's very safe, and it does produce a protection against smallpox. How well it works against monkeypox, we don't know in clinical studies because it's really only been inferred from non-human primate studies, and the non-human primate studies are a little challenging because the non-human primate studies were done with aerosolized and IV challenge as opposed to mucosal challenge, which is how it's being spread currently. And you might say, well, but mucosal isn't that better than intravenous or aerosolized? No, it actually turns out it might not be, because these mucosal lesions have probably an order of magnitude more virus than were given in the challenge study. So it's a real challenge here to deal with this. The majority of vaccine, I can tell you, that's been deployed in the U.S., although the U.S. government sent out a few hundred doses of ACAM 2000, the very large majority of vaccine that's out there by orders of magnitude is Jynneos. Initially, it was given via the subcutaneous route. Now it's being given mostly by intradermal route, although to some, that is children, and to those who might form keloids, or some immunocompromised individuals, it's being given subcutaneously like it was previously. I don't think there's much concern about a donation from somebody who's received Jynneos, whereas the issue is with if it became necessary to use ACAM 2000 widely, for at least 8 weeks after somebody gets ACAM, you probably would not want to be using they will have a virus potentially circulating, so you want to be careful there. And we'd really rather not have problems with needing to use vaccinia immune globulin to deal with those issues.
In the setting of cord blood banks taking donations, I think that just medically, many of us, I'm speaking without knowing, but would have made the decision to defer donations from donors who had the live smallpox vaccine for at least 8 weeks, but not for the killed monkeypox vaccine. Does that sound like?
Yeah, that sounds right. It's not, and remember, it's not killed. It's just it can't replicate.
Yeah.
But it's the equivalent. It's the equivalent. It's the equivalent. I don't know. It's because we always argue whether viruses are live or dead. Even if you consider it, it doesn't replicate, so it's the equivalent of dead.
Okay.
But we could argue over that, but let's not.
All right.
But that a little bit spills in because, currently both for cord blood and other, we just asked about smallpox vaccine, but really now we need to know which one they got. And it's good to know that most people are getting the Jynneos because that's a much safer profile.
Yeah, again, I'm not really, Beth, I wish I could tell you it was zero, but I'm not aware of I know that at least the last I checked, I was not aware of any of the ACAM 2000 that had actually been put in arms in California or New York. And I can't speak to the entire country. This is a vaccine that's still used, by the way. It's a licensed vaccine. It's still used because our service members who are deployed to the Korean Peninsula, for reasons that you can just imagine it, they are still vaccinated with ACAM 2000 because of the higher risk of exposure. Otherwise, we really don't. It's not a vaccine that's used. It's mainly kept in our national stockpile in the event of an emergency because we really hope never to see smallpox again.
Dumb question, but what's the expiration of those vaccines? What's the shelf life?
Oh, wow. Really good. It turns out, great question. It turns out that amazing smallpox, these orthopox viruses, the modified vaccinia Ankara, the non-replicating version, in our stockpile has about a 3-year lifespan. That's because it's stored at -20. When it's in Europe, where they use -50 storage, it's good for 5 years. And if it's stored at -80, it's good for 10 years. It turns out, though, that the majority of our stockpile in the United States is actually ACAM 2000. And that turns out to have a very long shelf life, like a decade plus when you go back and test it. It turns out that that smallpox itself is remarkably robust. And though I can tell you this from an experience we had, this should not have happened. But we accidentally, when we moved our laboratories from the NIH campus to the White Oak campus, we accidentally found some vials of smallpox in a refrigerator. In a refrigerator, that is, not in a freezer.
Wow.
And 45 years after they were put there, they were still viable. So we know that these orthopox viruses are pretty resilient. And actually, that actually is consistent with the fact that occasionally when they've found essentially human remains in permafrost, you can occasionally recover this as well. So again, robust, some viruses are very robust.
Wow.
That was probably trivia you didn't want to know either. So
no, it's
sorry.
good. Trivia is fun, as long as you learn from it.
Okay. I was going to change subjects. I was thinking another question that came in from a CBA member is that will manufactured cord blood-derived cell therapies using expanded human stem cells in lieu of transplant be regulated more like 351 cell therapy products or more akin to cord blood transplant BLA products?
Yeah, I mean, that expansion is probably going to be something that will be more like a 351 product. Obviously, we will try to make things, when we can find ways to streamline things, we will. But because of the expansion and the potential risks inherent in expanding things in culture, et cetera, it's probably going to have to be more like the 351.
Just a generic question, does time of expansion matter at all?
Yeah, it's turned out that what we've learned from work that's been done in our own labs and those of others is that it seems like even with one or two passages, you can potentially fundamentally introduce new risk into a product. So I'm not sure that whether it's in the incubator for 2 days or 2 weeks or 2 months, I'm not sure whether we can just give it a pass just because it's 2 days instead of 2 months.
I have a related question because something that I think we struggle with is when you use closed systems such as the Prodigy or the Quantum, and one is like we're always struggling with the GMP kind of ISO environment those need to be in, but it also what, again, that regulatory pathway is for those products as well.
Yeah, so Beth, this is a really good, so this is one where this is another TBD question in terms of we currently have a regulatory pathway which we're abiding by, but there are discussions ongoing about how best to regulate some of these devices that produce point-of-care biologics. And I don't think we have the perfect answer just yet because our goal is to make sure that at the end of the day, everyone who receives a product gets a safe and effective product. And we want to do so by the least burdensome method. Mm-hmm. And it's a matter of understanding what are the quality controls that need to go into this when you have a point-of-care manufacturing device. So there's a lot of discussion going on right now. And I don't want to jump the gun and say anything because I'd probably be wrong, but just so you know, people do realize that this is an issue that's coming up increasingly because by our own desire to see more of this, right? We are actually funding some research into devices that might help in the automated manufacture of gene therapies, right? And so this is since we are potentially going to contribute to this problem. We want to also help contribute to the solution.
Can you also add to your list that point-of-care administration of centrally manufactured products where the point of care is, say, a clinic, not a hematopoietic cell transplant program, but, an orthopedic clinic, as an example, where they have really no experience handling frozen products or, cell products. It would be great to figure out how to interact with practice of medicine in a reasonable way. I mean, you can't make a clinic room GMP, but how do you do that, safely deliver the product, and where can those guidances come from? Right.
No. Points well taken, and that may actually have to do with point of care devices and how do you make sure you're comfortable with how they're getting applied. Point well taken.
So here's another question from the community. With the boom in cell and gene therapy, the globalization of these treatments is necessary to ensure that all patients can benefit from it. For those that have used non-U.S. or ineligible starting material in the development and clinical development, what routes are there for FDA licensure or use for U.S. patents? So it's about starting material, and really, does it have to be GMP? That's
yeah. So there again, I think this is one where we need you. It really depends on the specific circumstance on where it's going. So I would think that for that person and that product, it's worthwhile coming in and having a discussion with the agency about this, because it will depend on our risk assessment of the non-GMP material that's starting and our assessment of what it ends up, what it goes through between where it starts and where it ends. So it's worth a discussion.
Can you talk a little bit about FDA's thinking around requiring tests for adventitial viruses, both in vivo and in vitro, or either or for manufacturers?
Yeah, so I think this has actually come up in a variety of settings. Most recently, and perhaps most pressingly, for allo cell lines in the CAR T cell world, potentially, where there are a lot of viruses. When you do PCR, you can find them, but what do they mean? And I think we're struggling, just as the rest of the community. What does it mean if you can PCR up HHV6, HAV7, a variety of these? Because in some of them, it's almost impossible to find somebody. You end up with a very small donor pool. So we are working our way through this. Again, I wish I had the final answer, but I think we're going to have to probably find some balance of understanding what is clinically relevant or potentially clinically relevant, and what is so low in copy number as to be not clinically relevant and potentially distracting from trying to have a donor pool. Again, because we do know that for some of these viruses, if you just do enough cycles of PCR, you'll find them everywhere. And so if you set your specification too narrow, you just end up having no donors. So we're working through this. I think part of this is that we keep finding new viruses that we're dealing with and having to work through them, but we'll get there.
Okay.
I guess similarly, so we perform gram stains. It doesn't, I understand, like any product that's not frozen, immediately thawed, and then administered. So if there's any time in between that, I don't know exactly what it is. I don't know if it's more than 24 hours or whatever. We perform a gram stain before release, and the gram stain inherently has a lot of false negatives, but it also has a fair amount of false positives. And a couple of times we've struggled with how, those products aren't administered, but I just was wondering if there was any evolved thinking on moving away from a gram stain.
Interesting that you mentioned that. There's discussion.
Oh.
I don't, again, I'm sorry.
But I'll take a discussion.
There has been discussion. There's been discussion, but I don't think we have an answer yet.
Well, at least it's appreciated.
Yeah. I mean, if you're old enough to have done them in medical school yourself, I think you appreciate the...
So I'm old enough to have, when we used to want the gram stains on sputum to dry quickly enough, you know how you make your own alcohol burner? Thank goodness they had the metal wrapper, the aluminum wrapper around the alcohol pad, so you could use that as the thing to set the little alcohol pad on, so you could light that, right?
Yeah.
I'm probably going to get arrested by the state of New York for having created a fire hazard at Bellevue now, but Yep.
Yeah. All right. Well, we're very close to the hour, so I want to thank you from all of us for giving us this time and sharing your wisdom and for your talk and for being available to us this way. It really means a lot, and it's really great. So really, thanks from all of us.
No, and thanks to you and to Beth. I think I actually have noted down some of the questions here because I think some of these are really good ones for us to make sure that as we go forward over the coming months, either in our guidance or in some of our statements, we try to address some of these things. And also very happy to entertain some of these conversations to try to avoid shortages in the cord blood world prospectively. So thank you.
All right, that's terrific.
Thank you very much.
Please go on.